Great. Okay, so I think we can get started. So good morning, or I guess afternoon or evening, wherever you're tuning in from. So on behalf of the co-organizers, I just want to welcome everyone to the Myeloid Network Seminar Series. Today's seminar is the 19th month of the program, so more than a year and a half of these successful talks bringing people together. We just wanted to thank everyone for their participation and both the participants and the speakers. It's been a really incredible platform. Just a reminder that next month we won't have a seminar because of the SITC annual meeting. We hope to see everyone in Boston for the SITC meeting. And then we're starting to plan our annual retreat, so save the date for March 9th, 2023. So I'm really delighted to introduce today's speaker for our seminar, Dr. Mikala Egeblad. Dr. Egeblad is a professor at Cold Spring Harbor Laboratory. She is the co-leader of the Cold Spring Harbor Laboratory Cancer Center program on cellular communications in cancer. Dr. Egeblad also serves as the chairperson for the American Association for Cancer Research Tumor Microenvironment Working Group. Her postdoctoral training was performed in the laboratory of Dr. Zena Werb at UCSF, which is where she began developing intravital spinning disk confocal microscopy to understand how the TME influences tumor progression. Her research group now studies how the tumor microenvironment contributes to therapy resistance and metastasis, primarily focusing on neutrophils and macrophages. Dr. Egeblad is extremely accomplished and has received a number of awards, including the Department of Defense Cancer Research Program, Era of Hope Scholar Award, the Pershing Square Sohn Prize for Young Investigators in Cancer Research, and the Suffrage Science Award. So Dr. Egeblad, we're delighted to have you here today, and so I'll turn it over to you.
Thank you so much, and thank you for organizing all of this. I really want to thank you for putting this program together. It's really been great to following, connecting with colleagues during COVID and beyond. So what I'm going to talk about today is primarily neutrophil extracellular traps. So we call them the dark web of cancer, and our insight into this comes from our interest in metastatic cancer. It's the worst type of cancer, of course. We have, as you can see, for most cancer, fairly good survival rate, 5-year survival for breast, prostate, lung, colorectal. But as soon as you will have a distant metastasis, your survival rate drops down. And metastasis is a very complex multi-step process. It's a process I like as a cell biologist. It starts with cells invading into the extracellular matrix, getting into the vasculature, surviving in the circulation, and getting out again on the other side, forming micrometastasis, and then actively growing metastasis. So my lab have been focusing on this process in many different ways, primarily in the end of the metastatic cascade. But today I'm going to talk about a very old connection to metastasis. So this is an example from a paper from 1978, showing that there is a relationship between the occurrence of necrosis or the presence of necrosis in the primary tumor and later risk of recurrence, metastatic recurrence, and also reduced 10-year survival. So you can see this here, the incidence of positive nodes is significantly increased if there's necrosis compared to if there's no necrosis. So why is there this connection between necrosis and metastasis? It's a little bit of a conundrum, particularly because when we think of necrosis, we think of it as a passive process that really is established as tumors are growing and outpacing the vascular expansion that is required for them to continue to achieve nutrients in the core of the tumor. So I had a new postdoc, Jose Adroa, that joined my lab just as COVID-19 pandemic started, and he started taking a new look at our tumors, starting with the classical 41 breast tumors. And what he noticed was, of course, there's a lot of necrosis in these tumors, but the necrosis didn't really fit the cartoon version of how we thought necrosis would look like. You can see here that they are very pleomorphic regions. And So it's not a core. There's these fingers coming out, and in these areas of necrosis, of course, there's a lot of neutrophils and a lot of neutrophil extracellular traps, NET or nets. So we are interested in nets because it's one of the 3 ways that neutrophils can combat pathogens. There's a classical phagocytosis, there's degranulation, and then there's these neutrophil extracellular trap formation. It's a process that is mediated mostly, as you will see, by PAD4-mediated citronation of histones. So it's one of the key signaling pathways in net formation, leading to chromatin decondensations, and then enzymes are associated with the chromatin, with the DNA strings, and the whole thing is expelled. So you recognize the nets by looking for DNA colocalized with granular enzymes and with the citronated histones. And this is an example from a COVID patient of what that would look like, where you have a myeloid peroxidase here on the extracellular lightly stained DNA, where you also see the citronated histone. Why would neutrophils have these peculiar methods of capturing pathogens? This is a movie we took on the spinning disc confocal microscope mentioned. You can see a lot of bacteria here, and in a moment a neutrophil's coming in, and it is perfectly capable of engulfing and starting to digesting these bacteria. However, sometimes you have too many bacteria, or in this case, you have pathogens that's too large. This is an aspergillospores. It's a movie from a group at Max Planck. And you can see that these neutrophils, there are about 5 of them here trying to gang up on the spore from all ends. And of course, the spore is bigger than they are. So this neutrophil is now condensing. Then you see the plasma membrane swelling up. There's propedium iodide in the culture medium, and you can see now the DNA is expanding and covering the spore. And so this is a way of extracellularly digesting the spore. So we like to think of this as sort of a Spider-Man that's casting a web to try to capture the bad guys, but unfortunately committing suicide in the process. We've been interested in nets and cancer for a while. The interest in nets started with work shown actually by Denise Wagner. I saw that you're on here, showing that there's thrombosis formed by nets in cancer. It was later shown also that the nets can capture circulating cancer cells and help them extravasate. And then since then, there's been a variety of papers showing that they are present in human tumors, that they correlate with tumor states and poor prognosis, that they can directly promote migration and invasion. They can promote the extravasation, as I mentioned, by capturing the cancer cells, but also by likely causing damage to the endothelium. And now there's new literature suggesting that they can inhibit anti-cancer immunity, and we and others have shown that they can support a metastatic niche. So we were really interested in nets, and Jose went back to these necrotic tumors and took a closer look at where they were found. And what he noticed was that not only were the nets present here in the necrotic regions, but when you look carefully at the vasculature, like this close-up here, you can see that there are aggregates of neutrophils, and these neutrophils are in the process of forming nets. They're positive for citrulated histone. He looked further using intravital imaging. So this is a movie where he labeled the vasculature with fluorescent lectin. So you see the vessel here, you see a region here with a lot of necrosis and infiltrating neutrophils. And then he labeled the platelets with a fluorescent antibody. And you can see how platelets and neutrophils are aggregating in the vasculature of these tumors. So Jose formed the hypothesis that maybe what was happening would be that these aggregates with the platelets were forming clots inside the branches of the tumor vasculature, and that way causing a lack of blood flow, and that would cause necrosis. And you can imagine that would be consistent with this pleomorphic necrosis that he was seeing. He looked using 3D cleared tissue stainings, where he then had vessels labeled here in green. Lectin was now stained in red, so you can see where there's perfusion. It only stains what's perfused, and then you see the neutrophils in cyan. And as you zoom in here, you can see again one of these intravascular clumps We have lectin here, and then the lectin stops, and we have a vessel that no longer is perfused. So it's very consistent with this idea that the necrosis is a process that's driven by these clots and a lack of perfusion. So Jose then went back to different mouse models to see how common is this actually. So we were expecting central necrosis, but you can see here he looked at 6 different mouse models of metastasis. And only the MDM-TV polyoma middle T model had this central core necrosis where there's not a lot of neutrophils and nets. All of these other models had the pleomorphic necrosis with the intravascular nets and with the abnormal vasculature. So that allowed Jose to start to look at the neutrophils in these different models and try to compare and contrast. What he discovered was, when you see here in the naive mice from the 41 background, the belt C versus the tumor-bearing one shown here for blood, but he's seeing it in multiple tissues. You can see that, of course, as we all know, there's a huge expansion of the myeloid compartment of the white blood cells. And if you look more closely on these myeloid cells, you can see that there are lots of neutrophils. As we target them, we mark them with Ly6G. Most of them are expressing Ly6C2 from high to intermediate levels, but there's also this population that is not expressing Ly6C. This population, interestingly enough, and we just started looking at that, also express CD11C. In contrast, in the polyoma middle T, like leukocyte population, we are not seeing this same extent of accumulation of the myeloid cells. And when you look specifically at the Ly6C marker, so shown here by flow cytometry, you can see that in the naive mice, again, we have Ly6C high expressing neutrophils. In the 41s, we now start getting the intermediate and the low Ly6C expressing neutrophils. But in the polyoma middle T, these neutrophils do not appear. They do appear in the Lewis lung carcinoma model, as well as the other models just listed. So that allowed now Jose to take the neutrophils from these animals and to sort them out, high versus low Ly6C, and look at what genes were expressed. And so this is a list of some of the downregulated and upregulated genes. And when he went through a pathway analysis for what kind of signatures this would represent, you can see that what is really coming out is leukocyte migration, cell motility, cell migration, migration, motility, migration, and also extravasation is showing up as some of the altered pathways in these Ly6C low neutrophils. So to look more closely at that functionally, Jose then used the classical chymosan-induced peritonitis model. So you're injecting chymosan into the peritonitis and then looking at how that attracts the neutrophils. You can see in a naive mice, again, we have the Ly6 high neutrophils, and that's in the blood. And after peritoneal lavage to see what's coming out, this is what's coming out, not surprisingly. In the tumor-bearing mice, in the blood, again, we have these 3 populations, the low, intermediate, and high. But in the peritoneal lavage, it's really only the intermediate and the high that are coming out. The low are not coming out. And that's also quantified over here. So we call them vascular restricted. They're not very good at extravasating even with a strong stimuli like chymosan. If we look at their ability to form nets, remember we were seeing these nets in the vasculature, something very interesting came up. So from the tumor-bearing animals, these Ly6 high neutrophils are not very good at forming nets using a classical inducer of net formation, PMA. However, the Ly6C low even have a higher spontaneous net formation after being taken out of the animals, and net levels more than doubles when you stimulate them with PMA. So the reason that you have the Ly6C low neutrophils, they have this increased net formation ability, or they're vascular restricted. And then in these pleomorphic necrosis region, we have, again, intravascular nets in these clots and aggregates. We have a blockage of blood flow. So to try to understand whether this was linked through the net formation, Jose then used PAD4 knockout animals, or conditional knockouts lacking PAD4 specifically in the neutrophil population. He inoculated these animals with the Lewis lung carcinoma, and you can see here in the wild type PAD4 animals, there is again a pleomorphic necrosis. You have net formation in these regions, and you can also see that the blood vessels simply are starting to disappear in these regions. In the conditional knockouts, there are still neutrophils coming into Lewis lung carcinomas. They're not forming nets, and you can see there's no major holes in the vasculature. There's no damage to the vascular network and necrosis. When we look at the tumors in a larger number of animals and quantify it, you can see actually already when you open up the animals that the necrotic appearance is very evident here compared to the conditional knockout for PAD4. It's also evident on histological slides. And so, of course, Jose went forward and quantified the amount of necrotic tissues from these slides. So you can see it's significantly reduced. We also have a reduction in metastasis in these animals. And interestingly enough, the mean area of the metastatic lesions is not altered. So what we think is going on is that we have fewer cells that are able to successfully form a metastasis, but once they're forming metastasis, they grow just as fine, reflected in the same area, same size of these metastatic lesions. So we now think we know why necrosis is happening in these models. It's through these intravascular clumps, but why is netosis, is this necrosis region associated with metastasis? So we think about it, we have the necrotic region, so these cancer cells are dead, they're not going to metastasize, but then there's a region right next to it, the perinecrotic region. And so we were very interested in trying to understand whether this perinecrotic region had some sort of selective pressure or other selective mechanism compared to the non-necrotic regions that would be responsible for these differences. So to look at that, Jose started using the spatial transcriptomics technology. So these are 3 examples of tumors he looked at. So this is the H&E staining before subjecting them to the spatial transcriptomics. And this is the unbiased clustering that he was able to do based on the transcriptomic analysis. And so you can see you're actually very clearly delineating the necrotic regions here in red and in dark orange versus the perinecrotic in orange and yellow, and then the green is the non-necrotic regions of these tumors. When he put together these transcriptomic analysis from multiple tumors and start analyzing them again, what is coming up in the pathway analysis is really 3 major things. So hypoxia, not surprisingly, is showing up as enriched in the perinecrotic regions. He's seeing EMT, TGF-beta production, TGF-beta signaling pathways, and then also positive regulation of EMT, epithelial to mesenchymal transition. And then he's seeing altered migration pathways. Just some examples of some of the genes that are altered is twist. So you can see twist is up here in cluster 2 and cluster 5, which are the perinecrotic regions. We have a CSF3 that's also high in these regions. Vimentin is high in these regions. And then interestingly enough, given that we came from the neutrophils, CXCL2 and CXCL1 are high again in the perinecrotic regions. So this is RNA, but of course we also wanted to look at the protein. And if you see here, after staining for E-cadherin and vimentin, and he's done that in multiple tumors, it's almost a black and white line that's going through the tumor where you have the perinecrotic region and where the cancer cells stop expressing E-cadherin. And then they start expressing higher level of vimentin, as you also can see here in the single channel slides or single channel stainings. So what we think is going on is that these tumors are sending out signals to the bone marrow, altering the myeloid poiesis. I didn't have time to show you some of the analysis Jose had done, but there's clearly skewing, of course, and altered maturation of the neutrophils. We are generating these Ly6 high neutrophils that are vascular restricted. When they get to the tumor, they are forming clots, and we believe that they are forming then particularly regions where the vasculature are poorly covered by pericytes. And so now you have the net formation where the platelets and the nets are clotting together. It's ending up blocking the blood flow into the tumors, and you have generation of this pleomorphic necrosis and the perinecrotic region. In the perinecrotic region, of course, we're seeing hypoxia. We think hypoxia leading to upregulation of TGF-beta and EMT pathway, and that is driving invasion of these cancer cells out of the tumor and allowing them to better metastasize. So the implications of this, I think, is first and foremost what this data shows is that tumor necrosis actually can be an active and untargetable process, something that we can try to target. So it's not necessarily a passive process. What we don't know yet, haven't fully proven, is whether this net-induced necrosis causes a metastasis via EMT, or if these are two separate things, EMT and metastasis, both the effect of nets. And Jose is looking at that right now. We wonder whether these type of neutrophils could be linked to what is known to occur in many late-stage cancer patients, where up to 20% of late-stage cancer patients actually are dying from clotting abnormalities. And it is known that some of this is related to nets. So do we actually see these vascular restricted neutrophils in humans? Do we see them in other organs in late-stage cancer? That is some of the work we are undertaking right now. We, of course, also see net-associated blood clots in other diseases, notably, for instance, COVID-19. So we are trying to expand a bit and see, are these type of neutrophils also present in other types of diseases or pathologies where you're seeing clotting involving neutrophils and nets. And so finally, of course, you may wonder, do these nets play a role in tumors where we are not seeing Ly6C low neutrophils? I showed you in the beginning that we have polyoma middle T, for instance, that are not having Ly6C neutrophils. So what's happening with them and with nets? And our insights into that work came from what I would call a rather stressful event in my lab. We had some results that were irreproducible. I had a graduate student in my lab that had been working on a project looking at cancer cell expression of CCR2. For years, she had shown over and over again that when the cancer cells were lacking CCR2, they grew very poorly until they finally escaped. And we were pretty sure we knew what the mechanism was. We were doing the last experiments, or by we, I should say, my unfortunate postdoc, Su-Yang He, who was tasked with doing the last experiments in this project. But as she repeated the core finding of the study, you can see here that in fact she would either find that the cancer cells lacking CCR2 had no difference in growth compared to wild type, or that in some experiments they were even growing faster. So we put our heads together for the longest time, 10 months actually to be exact, where we were going through the protocol to generate these animals line by line and comparing what could be different. Could it be the way they were isolated, centrifugation time, were they cold, were the matrigel the same, or something different with the way that she was dissociating the cancer cells. And finally, after 10 months, we came up with yet another thing that we wanted to try to troubleshoot. I don't remember what it was at this point, but I do remember Su-Yang leaving my office and saying, I'm going to order the mice. They'll be in next Wednesday, and then I'll start the experiment Thursday. And then I said, oh no, because we're getting our animals from Jackson Lab. It's about a 10-hour drive from Bar Harbor to Cold Spring Harbor. And they're coming in these boxes. And what I had failed to teach Su-Yang was that they needed to acclimatize. The mice, when they arrive here, look, we think, a little bit like this. So Xu Yang wanted to see if this could truly be the difference. She looked at the corticosterone levels in the animals as they were arriving to our lab, and you can see here the newly arrived animals had significantly higher level of this stress hormone in plasma than did control animals that had been acclimatized for 3 weeks. She tested also whether this had an effect on tumor growth. And you can see here that indeed, if mice are coming straight from the truck in these experiments, injected side by side with the same cancer cell population, they have a twice as fast growth rate. So Xu Yang was able to finish the project, reproduce the experiments, submit and get the paper accepted. But then we recognized, of course, that this could be really interesting to understand in more depth what was going on. Physiological or psychological stress has been related to poor cancer prognosis. It's a very nice review here from 2008 that's summarizing some of the epidemiological evidence linking stress-related factors to a higher incidence of some types of cancer, as well as a poor survival, and specifically a link to higher risk of metastasis. Conversely, there are also strong studies suggesting that social support for cancer survivors can significantly prolong their survival. So how do we mechanistically explain these observations? The stress circuit is a process by which the brain are recognizing some sort of stressful event, and cancer, of course, is very stressful for the patient and for the family. We don't know, as a patient, you don't know how your tumor is going to respond, whether it's going to come back, what is your next test results going to show, is a tumor back? And then on top of all of that, there's navigation of treatment plans and here in the States, at least, insurance issues. So stress is sensed by the brain and the hypothalamus, affecting the pituitary gland, and the pituitary gland is then signaling to the adrenal gland, causing the release of stress hormones, adrenaline and glucocorticoids. We thought we would study this in more depth, and we went together, started to collaborate with a neuroscientist here at Cold Spring Harbor, Linda Van Elst, that's been studying chronic stress in relation to depression and anxiety. So one of the classical models for this is the physical restraint model. It's very simple. As you can see, you take a Falcon tube and literally make holes so the mouse can breathe, and then you put the mouse in there for 2 hours every day. If you do that, you can see that the corticosterone levels are increasing over time compared here to control non-stressed animals. So we took polyoma middle T animals. This was the same model that had been used for the non-reproducible or the trucking experiments. And you can see here that if the animals are starting to be subjected to the physical restraint 2 hours every day, starting at the time that the tumors are palpable, there is indeed, again, an increase in growth of the primary tumors, but also in the metastatic burden that we're seeing at the end point. So this model looked good, but we wanted to expand to additional models. And so more recently, we've been working with Linda Van Elst and her group to use the chronic unpredictable mild stress model, or the COMS model. This model is a model that's also been used to study anxiety and depression. And it's good because it's a model where you can study resilience. The model is one where you now take a random stressor every day to the animal. So it can be food deprivation, it can be restrained again, but for a shorter time. The cage may be wet, the cage may be tilted, there can be light turned on during the night for the animals, stroboscopic light, white noise, owl sounds, or even fox urine. And so we subjected the animals to this random protocol. You can see here that, interestingly enough, we have one cage that are just not responding with any metastatic lesions, but 2 of the cages we are seeing a robust increase in the metastatic lesions, just like we saw with the restraint stress. we like this model because we can now start to do behavioral studies as well as more physiological studies looking at the hormone levels in individual animals. And one of the things you're seeing here is that these animals that did not have increased metastatic lesions also were the animals where there was no increase in corticosterone levels. So the corticosterones, we then started to look more closely at, specifically the glucocorticoids. It has been reported previously in breast cancer models that glucocorticoids can promote metastasis. So Yang started using corticosterone pellets rather than stressing the animals, and now using a model that we think is a little bit more reminiscent of what's happening in patients. So she's first inoculating the tumor cells into the mice, allowing them to grow sort of like what happened in a patient that may not know that there is a tumor growing. The mice then undergo surgery to remove the primary tumor, and then the pellet, either vehicle pellet or corticosterone pellet, is inserted. And finally, after 77 days, the lungs are taken out for analysis. So what you see here is that there is a huge increase in the metastatic burden in these animals. We do find single cells that have disseminated with a vehicle pellet, but there are not as many of them go on to form successful large metastatic lesions. As shown here, you can see the number significantly increased and the overall metastatic burden is increased. So to determine whether or not the glucocorticoid receptor was acting directly on cancer cells, which do express the glucocorticoid receptor, the next thing Su Yang did was to knock out the glucocorticoid receptor in the cancer cells. So she have generated with other people in the lab a cell line for the polyoma middle T mouse model. And she used CRISPR-Cas9 to knock out NR3C1, which is the gene encoding for the glucocorticoid receptor. But you can see when these mice are subjected to stress, there's no decrease in the number of metastatic lesions, no decrease in the overall metastatic burden. So it really doesn't matter. These cancer cells are still forming metastasis in response to stress. So that, of course, led us to think it had to do something with the microenvironment, and to look more at what is the effect of stress on the environment and the animal. Su Yang took cancer-free animals, so now there's no cancer, and it's just a stress exposure for 3 weeks that she's looking at. And you can see that stressing the animals daily leads to a huge accumulation of extracellular matrix, shown here for fibronectin, in the lungs and also in other tissues. There are fewer T cells that now infiltrate the tissue, specifically where we see a reduction in CD8 T cells. And then there's an altered chemokine environment, and we're focusing currently on the CXCL2 and CXCL5 chemokines that are upregulated in tissues, or specifically here the lung, from animals that has been subjected to chronic stress for 3 weeks. So CXCL2 and CXCL5, of course, are interesting to us because they are capable of recruiting neutrophils. So Su Yang looked for the neutrophil infiltration, and as you can see, it is significantly increased in animals that have been subjected to restrained stress. So do the neutrophils contribute to this stress-induced metastasis phenotype? To test this, Su Yang used a depletion model, depleting the animal with a Ly6G antibody. She here used an experimental metastasis model because we know that the depletion is not going to be so long lasting. It did last for the 21 days, but we wanted to narrow the experimental time so that we could ensure that there were no neutrophils present. You can see here that compared to IgG control animals, again, we see increased metastatic burden when the animals are stressed. If the animals are depleted of neutrophils, the metastatic burden and the number of lesions is significantly decreased. So the neutrophils, again, are pro-metastatic also in this model in the context of stress, but how do they do it? What does stress do to the neutrophils to make them pro-metastatic? So to look at this, Su Yang have been looking at neutrophils from stressed animals and also neutrophils in vitro, where she isolated them from the bone marrow and then stimulate them with various glucocorticoid receptor agonists, including in this case, that's a GSK agonist. what she's seeing is altering in a variety of different genes, and specifically we see changes in the circadian clock genes such as PER1 or CRY. She sees changes in genes involved in migration and inflammation, and then a lot of big changes in oxidative and antioxidative genes. Many of these genes are direct targets of the glucocorticoid receptor. It's transcriptionally involved. You can see here that the receptor is associated with the locus of CBTB16 of MAP3 kinase and PER1, one of these circadian clock genes. So the circadian clock gene connection led us to think more about the circadian rhythms in the neutrophils. So many of you are probably aware that the neutrophils, as well as most other immune cells, go through these circadian rhythm changes, where they are changes in the number of cells that are present in the peripheral system in the blood. So you can see that the neutrophils are released towards the end of the active phase in the animals. Then they start to be cleared, and so there's this cycling of total number of neutrophils, and there's of course also a cycling in how many of these neutrophils are freshly released from the bone marrow versus those that are aged. So Jose and Su Yang started looking at whether or not there were neutrophil number circadian changes in the stressed animals, and as you can see here, they still continue to cycle, but the amplitude of the cycle is smaller after the animals have been stressed, and also it's being changed. So the peak of the neutrophil numbers is now happening at an earlier time than it is in control animals. It's not just the number of neutrophils that changes, the gene that's expressed by neutrophils changes over the circadian rhythm. And so one of these genes, for instance, is CD62L. You can see again, when we look at the expression of CD62L, it changes over time with stress. So we are moving the normal circadian rhythm, and it's no longer timed to when the light is on, when the animals are active. So what kind of functional effects could these changes have? Well, one of the things that, of course, was standing out to us, particularly given what I just showed you, is that these more aged neutrophils or altered neutrophils have a propensity to form more neutrophil extracellular traps or nets. So Su Yang went back to look at the animals and to look at nets specifically in the plasma of animals that had been subjected to stress. And you can see that indeed in the animals that had been subjected to restraint stress, there was more than a 4-fold increase in the number of nets we could find in plasma using a sandwich ELISA assay. This was dependent on the presence of the adrenal gland. So if the adrenal gland is surgically removed, stress is no longer inducing nets. If you take out neutrophils in vitro and stimulate them with PMA versus with glucocorticoids, you can see that actually glucocorticoids here, the synthetic dexamethasone, but it's also shown here quantified for the endogenous corticosterone. You're seeing that these glucocorticoids are capable of inducing nets almost at the same level as PMA. So I mentioned previously and showed you that PAD4 is important for net formation. And indeed, when we look at PMA-induced nets, PAD4 is very important. However, in the context of these glucocorticoid-induced nets, they are really resistant to it. We don't see any difference with PAD4 inhibitors. So to look more at the role of glucocorticoid signaling in net formation in these stressed animals, Su Yang generated now conditional knockout animals that are conditional knockout for the glucocorticoid receptor in the neutrophils. So you can see here neutrophil expression of the glucocorticoid receptor is way down. And if she takes neutrophils from these animals, again, they are forming perfectly normal levels of neutrophils, of nets in response to PMA, but in response to dexamethasone, or as quantified over here, corticosterones, they are no longer forming nets at a high level. So in vivo, then, when we look at the ability of these animals to support metastasis, she took the conditional knockouts versus controls that were lacking the Cre, again, using this model where we are allowing the polyomyloid tumors to form tumors, spontaneously disseminate, and then surgically remove them, and exposing the animals to daily restraint stress before looking at metastatic burden. What you see here is in these animals that are conditional knockout for the glucocorticoid receptor and the neutrophils, stress really is incapable of promoting metastasis. And this is not the case if she does the same experiment now with conditional knockouts for the glucocorticoid receptor and the T cell population using the LCK Cre. So it is specifically in the neutrophils that the glucocorticoid receptor is so required for the promotion of metastasis. So what we've shown so far is that glucocorticoid receptor signaling in the neutrophils required for the stress-induced metastasis. It's required for net formation. But of course, are these two processes linked? And to look at that, what she did next was to treat with DNase 1. As I mentioned, we can't use a PAD4 inhibitor. So she was doing daily injections with DNase 1 to digest the nets. If she does that, you can see in the plasma, indeed, she hits the target. There are significantly fewer plasma nets. And in the metastatic burden experiments, you can see a very significant reduction in the animal's metastatic burden after treatment with DNase. So how are the nets working in this context? We think they're doing multiple things. So it's not just the nets themselves, but really the many different things they're having on the microenvironment in the lungs. One of the things we have looked at in more detail is the extracellular matrix accumulation. So as I mentioned, we have more fibronectin with stress, but when we treat with DNase, we do not see that accumulation. So in conclusion, as we all know, stress leads to the release of glucocorticoids. The glucocorticoids are having major effect on the neutrophils in the animals, leading to an abnormal maturation, abnormal circadian rhythms, and increased lung infiltration and increased release of net formation in a PAD4 independent manner. In the lungs that support the formation of metastasis, and we think it's doing that through multiple effects, acting on the extracellular matrix, on T cell inhibition, and on various other downstream pro-inflammatory mediators. I wanted to acknowledge the work by Dimitri because, of course, there's additional complexity. Here we are really focusing on the effect of glucocorticoids, but his group had a very nice paper a couple of years ago where they were showing that norepinephrine also are acting on the neutrophil population, in this case leading to effects and release of S100A proteins, MPO, and lipids. And again, showing that the effect of these neutrophils were PAD4 independent, acting on promotion of metastasis through FGF receptor signaling in cancer cells. And I think it's perfectly likely that multiple pathways are happening at the same time. We should not forget the norepinephrine. The implications of this, I think, is that we really, I think, should take stress very serious in the context of cancer patients and cancer survivors. And reducing stress or targeting neutrophils and nets may be a very important way of increasing survival for patients that already have disseminated cancer cells, and also means to reduce the pre-metastatic niches. I think we need clinical studies, and we are hoping to get deeper into at least the first kind of studies where we can look at net levels over time and correlate with outcome. The synthetic glucocorticoids that we also used in this study, of course, used clinical in cancer treatment. They're used for in connection with chemotherapy to help with nausea, as well as with various edema situations. Is this an issue? It's used a lot. I don't know that that's clear yet. There is one study that tried to do a meta-analysis of this, and they didn't really clearly make a conclusion on this. It's possible that it really matters what time of glucocorticoid that's being used, what effect it has on multiple cell types. But I think this again is something that needs to be looked at. I am also very intrigued about these results on the circadian rhythm changes, and I wonder whether there's any effects of these desynchronized neutrophils, whether that potentially could contribute to other types of physiological macroenvironment effect on metastasis, sort of as shift work, jet lag, and sleep interruptions, all of which is known to have a correlation with cancer progression. And I just want to highlight that we did not just look at the neutrophils and their altered circadian rhythm in response to stress, but also lymphocytes and monocytes. And so you can see all of them are altered, but they're altered in different ways. This is the neutrophils moved this way. The lymphocytes are also moved, but they're moved even more, and their numbers are generally reduced. The monocytes, the circadian cycle is almost the same, but you can see that the amplitude is much lower. So do we have a desynchronization of the different types of immune cells in some of these situations? I think that would be very interesting to try to study. And I want to say that I think the stress biology is really a nice area to start looking more deeply in. I showed you here today that it has effect on tumor growth, on metastasis, and that could be links with resilience. We all know that different people respond differently to stress, as did the mice. So is this something we can try to understand at the mechanistic level? What I didn't show you yet is that we're also finding effects of stress on response to therapy. Here is some preliminary data on checkpoint inhibition. And you can see here that in this model that the checkpoint inhibitors are kind of capable of keeping the tumors stable, but if the animals are stressed, they no longer have any effect. And then, as I mentioned there, this effect on circadian rhythms. There are a lot of challenges in studying stress and cancer. I think it's really a topic that brings together the microenvironment with the macroenvironment, how the body physiologically responds to external stimuli. But some of the challenges are, for instance, how do we actually even define stress, particularly in people, and how do we define resilience? Developing better animal models, I think, is still needed. Getting good epidemiology, and that of course go back to how do we define and measure stress. And then I think it's an exciting area because it's really transdisciplinary. You need to bring together, I believe, people with expertise in psychology, epidemiology, neuroscience, immunology, cancer biology, microbiota, maybe even, and circadian biology. And so I just want to make a last plug for saying that we are looking at postdoc candidates that would be interested in going into this area. And these postdocs will not necessarily have to be awake all night to take all of these samples. Maybe we can divide it out a little bit, so it's not a 24-hour postdoc. And with that, I want to end by thanking the people that did this work, particularly Sue Yang, who went into the lab and stressed animals day in and day out, as you could see, as well as Jose, that helped us really start to understand the circadian rhythms, and also were very observant in looking at necrosis and understanding this new phenotype of neutrophils and its potential function in cancer and in driving necrosis. I want to thank my collaborator, Linda van Alst, helping us with the neuroscience of stress, Chris Barcock, that's helping us with understanding the transcriptional changes of glucocorticoid stimulation, and Doc Fearon helping us with all-type immunity. And with that, I'll be happy to take any questions. Thank you very much.
Thank you for two wonderful stories, very comprehensive bodies of work. So congratulations to you and your postdocs.
Thank you.
I have one very basic question about how you envision metastasis occurring if blood flow is interrupted by these nets.
So I don't envision it occurring from the regions where there is interrupted blood flow, but really those regions around. We think that they're being pushed to migrate to the areas where there's more, potentially more oxygen, more flow, and then get in that way. But it is one of the challenges, really trying to track the individual cancer cells and see how do they make it out of that perinecrotic region.
Yeah, great. I just wondered also whether you've ever experimented with artificially reducing oxygen to look at the effects on some of these systems that you described. Because if you're blocking, I haven't thought through this very well, so.
Yeah, no, there's a lot of things we want to do next in trying to target these pathways. We have started looking at the specific molecules involved in the hypoxic regulation and the link to EMT, and starting to knock out some key molecules in the cancer cells to look at that.
Okay, thank you. There's a please ask your questions directly if you like, but I'll read a couple of questions from the chat. Joanna Joyce, thank you for a great talk. And then she wanted to know whether you think your striking results can be connected to recent results from Mikala Egeblad's lab showing that circulating metastatic cancer cells increase during sleep.
That's very interesting. Possibly. So we want to do some experiments on that with altering the sleep in the mice, altering their circadian rhythm, and try to look at when the neutrophils and the cancer cells are coming out. Indeed, we have not done that yet, but I think it's possible that it's linked indeed.
So Sarah Mowbray asks, have you looked at peripheral blood mouse neutrophils versus bone marrow neutrophils, and do you see similar trends?
We have. So Jose has looked at the bone marrow quite extensively. I didn't put it in here because, of course, it's a little bit more complicated, but we are seeing the same trends indeed in the bone marrow.
Yeah. So Eric B asked, one of the unregulated genes that came up for Ly6C low neutrophils is Siglec-F. So he thought that Siglec-F neutrophils were long-lived, so their propensity to release nets more than Ly6C high neutrophils is paradoxical. Can you comment on that?
Yeah. I don't know. I mean, so they are forming a little bit more nets unstimulated, but we do think in vivo it's not only unstimulated nets. We are seeing them accumulating specifically in regions of the vessels where we are seeing extracellular matrix exposure. So we think that these neutrophils they may be more long-lived, but they're getting a stimuli that is triggering the net formation, and we think it's a poor vascular composition within the tumors that are triggering this.
Okay. Again, I want to remind people, please go ahead and ask your questions so you can have dialogue. Oh, Denisa. Hi. You're muted still.
Stress activates endothelium and causes release of things like von Willebrand factor and P-selectin, which recruit neutrophils and are also thrombogenic. So that's kind of interesting link to what you were talking about, and the microthrombosis playing probably a prominent role. In that same thing, hypoxia activates netosis and promotes thrombosis.
Yeah, I thank you, Denise. I think there's a lot of feedback loops going on here. And once you start having some of these first thrombi formed, you will get more and more. You'll get more hypoxia. We do see that, right? Once a thrombi start forming, you just see more and more forming around in these tumors. So I totally agree with that. We haven't looked for whether the stress is causing any thrombi in the tumors. because we have mostly focused on the stress in the metastatic side and the effect on the cancer cells when they're disseminated. But we probably should go back and do that too. Thank you.
I have a basic question. Are there circumstances where an increase in neutrophils in cancer is beneficial, or where nets are beneficial in cancer? Have you ever observed that, or is it always?
I don't think so. We had a collaboration when we thought so, and then the right experiments targeting nets were done, and then no. So I don't think so. I think that the effect on, I mean, particularly the intravascular thrombosis is just so dramatic that you don't want too many of them.
I just want to say, and we study various therapeutic approaches to inhibit mainly monocytes and macrophages. And one of the things we notice is there appears to be like a resistance that's the tumor cells change to produce much more neutrophil chemoattractant, so the neutrophils increase. So I would think that maybe neutrophils might be a common increase in neutrophils and nets might be a common resistance source for therapy, immune therapy. Have you ever?
Yeah, so we're starting to look at that now. We haven't. We don't really have any data on it yet, but it's ongoing. I think there is a lot of data showing that there's this balance between, with the macrophages are inhibited, they switch to neutrophils, and there are a lot of models really, either they're driven by macrophages or neutrophils.
Yeah, maybe I can show you our data and we can talk, because it's really interesting. There's some more questions here. From SS. When we use steroids to treat severe COVID infection, we may be they want to know, are they increasing intravascular nets and platelet aggregation and thromboembolism?
That's a bit of a conundrum for us indeed, right? Because you could worry about that. I'd love to look at it in patients. We know that the human neutrophils do respond to the glucocorticoid the same way as the mice. We've done those experiments, but we haven't been able to get neutrophils from COVID patients and see how they respond to the glucocorticoids. We know that they have more nets when they are severely affected. It's possible that it's related to when you give the glucocorticoid treatment, you have different effects. And of course, I mean, we have other cells and neutrophils. So the benefits acting on other cells may outweigh anything on the nets.
Yeah. Another question, do you think that your results could explain an article published in Lancet Oncology showing that patients treated with anti-PD-1 given in the afternoon are less likely to respond to treatment than patients treated in the morning? Wow.
I don't know of that specific study and specifically PD-1, but I do think that the timing of the day is going to be super important. If it's all neutrophils, who knows? But definitely, I mean, as I showed you, right, the lymphocytes are cycling too, the macrophages are cycling. So I think that the whole field of Sicaria and immunology is going to be super important in how we think of responses to treatment.
Those are all the questions in the chat right now.
Lisa, I have a model question for you.
Yep.
You showed early in your talk that the polyominomality and the C3.1 tag models are really quite different with their neutrophil content, net content, and some of the biomarkers, Lys6C, Lys6G, blah, 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 blah. So based upon that, I was surprised that you then went back to the PIMT model in your subsequent studies to look at the stress response in PIMT.
So we think in some of the other models, you have so many changes in the neutrophils and so high levels of nets already that the stress may not give so much extra, but additionally to that, We have the problem that not all mice models respond equally well to the stresses. So the BALP-C model, for instance, do not really respond to the stress inducers. They respond initially, the glucocorticoid levels go up and then drop down as they sort of like, okay, here they come again. They just get, they ignore it. And the black sixes do not, they tend to do it for a longer time. So it has a little bit to do with the black six model. It also had to do with the fact that, I mean, we had seen it in our sort of unintentional observation with the trucking of mice. That was a black six model. And then for the neuroscientists, they use the black six model a lot. So these are the different reasons why we've done it. We have looked in other mouse models. We have a pancreatic cancer model that also metastasized more when we subject it to stress, but it's also a black six model.
Mm-hmm. So a lesson in really knowing your model really, really well. Paying attention to what strain background your mice are on, and not coming to huge conclusions based upon one model on one strain.
Yeah.
Good lesson, I think, for all of the junior faculty and younger scientists that are on the call today.
But I think it's still, I mean, if you get a strong result in one model, I think it's still important, right? But you just have to think of it as mice are different and people are different, right? And we respond differently. Our genetic background's also different. So I think not necessarily having the same result in all the model is perfectly fine for me, as long as we're trying to understand why these respond this way and these in different way.
Really spectacular story. Congratulations to you in the lab. Really lovely.
Thank you.
Can I ask one more question? Since you've done this very beautiful, careful work, have you determined how long mice should be equilibrated before, so that they would rest before they have this stress response?
We have not done it systematically. We used to do just 5 days, and after these data, nobody does it less than a week. We know that 2 weeks is fine. I think a week is probably fine.
Yeah, you know, it made me wonder whether treatments like gavage and things, you know, that are probably stressful for the mice.
Super stressful, and I think it's important that you really give your control animals a vehicle gavage, because that is a super stressful event. I think we also need to think about the temperature in the animal facility. It's stressful. We need to think about the noises in the animal facility, including the owl sounds or cats or things like that. The music, if there's somebody is playing music when they're changing the cages, maybe that has an effect. There's even studies showing that mice respond differently to male and female handlers. So one really has to be careful with the controls.
Yeah, very good.
But it's, I mean, it only gets worse if we think about measuring this in patients, right? So.
Well, I'm full of curiosity, so I have one more question. You know, you showed this beautiful connection between necrosis and nets. So I wasn't sure whether this is the right conclusion. Do tumors that show necrosis then always have nets? Or do you find cases where there's necrosis and it's due to something besides nets?
Yeah, so in the polyoma, we see regions that are necrotic with no nets. But I think it sort of goes back to Denise's question. So once you're starting to have necrosis and hypoxia, there will be neutrophils coming in and they will form net. But in this case, it's more like a reactive net formation. We think that one that is necessarily driving it.
Okay. Yeah, wonderful. Well, thank you very much.
Can I just jump in to ask a quick question again, Mikala, if that's okay?
Yeah.
It's a very broad question. It's what's your advice for patients with cancer? It's one of the most, probably the most stressful event that you can deal with, a cancer diagnosis. Everybody says, you know, reduce your stress levels. Do you think that that's practical to do? Do you see that maybe in this situation targeting the pathways in neutrophils, maybe targeting the nets, could actually be a scenario where that's more effective. What are your, just very general feelings about that?
I think we still need better drugs, better tools to target the neutrophils to prevent this from happening. I mean, it's so early stages in developing drugs specifically to the neutrophils. So once they are there, I mean, that's hope, right? That you will be able to do this. Mm-hmm. I think it's possible that some of the neurologic drugs that are already used for depression and anxiety, they may potentially be beneficial. So that's certainly something that we will be looking into in the mouse models. But for now, I think the studies that's been done epidemiologically showing how social support can increase survival time, they're pretty strong in my opinion. And there's significant changes. The changes are very significant actually. So I would advise them to do the best they can in getting a strong social support, maybe beyond family. Even there's these social support groups that many of the hospitals are organizing, and I think there's a very strong rationale for really doing that.
That's interesting that you say the antidepressants, Mikala, just, kind of casually, but, in Joanna's recent paper with Doug Hanahan, they showed that a tricyclic antidepressant significantly improves inhibition with, I don't remember if it was PD-1 or PD-L1. And so they linked that to macrophage, transcriptional reprogramming, et cetera. Joanna, did you guys look at the neutrophils in those models?
So this work was really led by Aga, a PhD student in Doug's lab. We only helped very, very peripherally in that. I don't think that they specifically looked at neutrophils in that context, but they are looking at neutrophils in other contexts in that lab. Aga may even be on this call herself. If she is, then she can speak up. But yeah, that's interesting. Also, of course, antihistamines implicated from a completely different perspective to what we're talking about here. But I think it's really quite remarkable how many of these drugs that we don't consider at all in the context of cancer can potentially be co-opted when one starts to really go down into the physiology as well as the cancer biology. Mikala, can I just ask you one last question that's not related to the content of your talk at all, but this movie that you showed, it was absolutely amazing. Did you make that, or somebody in your lab made this?
No, I wish. Jose's partner made that.
Oh, it's wonderful.
Yeah, I think
congratulations to them. It's just, yeah, it's incredible how a movie can just make things so clear and also leave such an impression. I will never forget this. So never forget the first one of your talk, because that movie was incredible. Thank you.
Well, thank you so much again on behalf of all of us for the great talks, and look forward to hearing more from you in the near future.
Thank you. Thanks for having me.
Bye, everyone.
Bye-bye.
Bye.